Morning, everyone. It's a pleasure being here. Thank you for your time and attention today. I'd like to share a little bit of information about some new results that we're pretty excited about in myopia. Stuart's clinical stage company. We're developing first-in-class platform technology that's described as a collagen mimetic peptide. It's a fractional synthetic strand that mimics collagen in the body, and it's designed to repair collagen in situ, and has a number of different important health benefits. Our pipeline's expanded quite a bit since we spoke with you last year. Last year when we came to OIS, we were talking about our dry eye program and our glaucoma program. Now we've added new indication areas in GA and now myopia, and we're very proud of our new relationship with the Glaukos folks in neuroprotection and glaucoma. Everybody here has heard the story about how half the world's going to be myopic by 2050. That's going to be exciting, I think, for the industry, but I think the more important part is about 1 billion people around the world are going to be high myopes, and there's some serious problems with those patients, as you guys all know. Much higher glaucoma, retinal detachment, and other complications associated with those patients, and treatment options are pretty limited. Regardless of your theory about why myopia originates and what the signaling cascade is or what the challenges are associated with it, it always ends up resulting in dysregulation or challenges to the posterior collagen biomechanisms. We've got normal remodeling occurring as the eye develops and grows in the collagen tissue. That remodeling takes place, the collagen's digested, is replaced, and so forth. But in myopia, those mechanisms are dysregulated, and it results in an overall loss of scleral integrity, and that's what causes that change in the eye shape and the elongation and the axial length change. If we could repair that tissue in situ, we could change or halt the axial lengthening. Our peptide, our platform, repairs damaged collagen. That's one of the key elements of the mechanism of action. That MOA includes the repair of collagen tissues in membranes and structures. It also includes the reduction in inflammation signaling and reduction in oxidative stress. It's highly selective. It repairs collagen. It does not build up. It is not a cross-linking technology. It is, and we've demonstrated and proved that we only bind to damaged collagen. We'd done prior research in 2021 on cornea and lamina cribrosa collagen structures and demonstrated repair in those structures in the past, and we decided this year to sponsor some studies to look at the effects of CMPs on scleral tissue. We wanted to look at those. We wanted to look at both the posterior, look at the retina, Bruch's membrane, the choriocapillaris, the choroid, as well as the posterior scleral tissues. These are some images from that first set of tests that we did. And across the top, you see images of the virgin sclera. You see the cross-hatched look of the short collagen fibers and the long fibers in the scleral tissue. This is an ex vivo evaluation of rat sclera. Across the center, after digestion from matrix metalloproteinase, which we know is elevated in myopia, you see the massive deterioration of the short fibers in the collagen. That same tissue, once treated with the CMP, is shown on the bottom row, where there you see a restoration of that collagen tissue integrity, similar to the untreated tissue. We also, this is the second presentation where you hear about atomic force microscopy and Young's modulus in the sessions this week. We used AFM to do a number of measurements on the tissues and take a look at what's the strength of the tissue and measure very accurately, and AFM gives you thousands of points of measurement, which gives you a lot of accuracy from a statistics perspective. So in the center graph, you see the baseline of the strength measured by Young's modulus of the sclera. MMP digestion causes a massive reduction in that tissue integrity, and when we treated with the first shot of CMP on that same digested tissue without any sort of dose ranging or anything like that to evaluate it, we got a 23% increase in tissue integrity. So that was a very exciting outcome for us. It seems like the most direct approach to dealing with myopia-related damage. We target the damaged collagen in the sclera, and we have the ability, we believe, to reduce the rate of myopia development. Just to make sure that we were doing what we thought we were doing, we did some tissue staining, and we take a look at and stain for damaged collagen in the sclera and the glial lamina, And we noticed, you can notice in the diagram the dramatic reduction in the red staining on the right-hand side, which is indicative of a reduction in damaged collagen tissues. Safety profile is important for myopia. You're dealing with pediatric indication. It's important that it's comfortable, safe, and easy to use. The safety profile of CMP has been demonstrated in a number of different studies. We've done tox work in our dry eye program. The dry eye program itself saw a very, very positive safety profile in humans. And we think for myopic patients that CMP treatment is a substantial advantage over alternatives. We don't expect a rebound effect like you would see with atropine. We don't paralyze the focusing mechanism, so you don't have any accommodation required for children who are taking the therapeutic. There's no light sensitivity. We think we've already got a formulation that we know can get to back of the eye. We know that we've got a comfortable formulation as well, so this looks like it's going to be something that can really make great strides in this area. That target window at 4 or 5 years old or 6 years old to 18 years of age for patients as they develop myopia is a great range for us to operate. We think we can bend the curve downward in terms of myopia development. We've done primate testing demonstrating that we can apply the therapeutic topically, and it encompasses the entirety of the posterior sclera. So we believe that we're in a great spot here. We target the tissues implicated in myopia progression. We've got a demonstrated MOA in multiple areas of the eye. We've got a preclinical proof of concept in ex vivo tissue, strong safety profile, and we don't believe that we're going to have any atropine-like side effects. In a topical drop formulation, we think this is the way to go for myopia, and I appreciate your time and attention today, and I'd be happy to answer any questions after the session. Thank you so much.